Everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s Bionet. In this video, we will be talking about the next concept under molecular basis of inheritance, that is a search for genetic material. Today, we know very well that DNA is acting as a genetic material, and we know very well that this is involving in the transfer of genetic characters from parents to offspring through a process called reproduction. But just before 1940s, nobody knows that this DNA is acting as a genetic material. Nobody is aware of which molecule is involving in the transfer of genetic characters, whether it is DNA or protein or RNA or a lipid molecule. But by the year 1942, the two scientists called Hershey and Chase provided an experimental proof for the DNA is acting as a genetic material. Under the concept of the search for genetic material, we are going to discuss in detail about these three experiments. One is Griffiths transformation experiment. One is Griffiths transformation. In the year 1928. This experiment concludes that organism must have a transforming principle which involves in the transfer of genetic characters from one organism to another organism. By his experiment, Griffiths, he came to a conclusion that the transforming principle might be a protein, but actually protein is not the genetic material, but DNA is a genetic material. And next one is biochemical characterization. Biochemical characterization of transforming principle, Griffiths transforming principle. This was conducted by McLeod, Avery and McCarty, the three scientists, in the year 1938. This experiment confirms that the DNA is acting as a genetic material in the organisms. This is a completely in vitro experiment where they did not use any organism to say this organism is having a DNA as a genetic material. Because of this reason, most of the scientists did not accept the results got by McLeod, Avery and McCarty by this experiment. And the third one is Hershey and Chase experiment. Hershey and Chase experiment. This experiment provides an unequivocal experimental evidence to say that DNA is acting as a genetic material. This was conducted in the year 1942, where they did experiments on a T2 bacteriophage and E. coli cell. Before we are going to discuss in detail about these three experiments, first let me give you a brief historical background in search for genetic material. Earlier, the first attempts in search for genetic material were made by two scientists. One is Mendel in the year 1866. In the year 1866, he proposed the principles of inheritance and variation. Principles of inheritance and variations. Okay, by this he came to say that how the progeny carries the parental characteristic features, how the progeny of sexual reproduction shows variations. Okay, and next Mischer, Frederick Mischer, in the year 1869, he identified nucleic acids in the nucleus. Okay. To understand that the DNA is acting as a genetic material took a very long time of about more than 80 years. But in the year 1942, Hershey and Chase came to provided an experimental proof that this DNA is acting as a genetic material. In this long period of 80 years, some scientists like Morgan, Sutton and Boveri were made some attempts to know the genetic material, but their experiment in search of genetic material were narrowed up to only chromosomes. They could fail to know what molecule of nucleus is exactly acting as a genetic material. By the year 1926, most of the scientists have started their quest to determine the molecular and biochemical nature of a genetic material. Later in the year 1928, Griffith unexpectedly came to know that the organisms are having some biomolecules acting as a transforming principles or genetic materials that is involving in the transfer of genetic characters from one organism to another organism. Now let us move on to Griffith's transformation experiment in detail. You know very well that this experiment was done by Frederick Griffith in the year 1928 on Diplococcus pneumonia. Here the Frederick Griffith conducted a series of experiments on Diplococcus pneumonia. This Diplococcus pneumonia is otherwise called Streptococcus pneumonia. Streptococcus pneumonia. This Streptococcus pneumonia or Diplococcus pneumonia causes pneumonia disease in mice. Pneumonia disease in mice. He conducted a series of experiments on this Diplococcus pneumonia and finally came to know that there are two strains of Diplococcus pneumonia. One is S3 strain of Diplococcus pneumonia. This S3 strain is a capsulated strain and is virulent strain. Virulent or pathogenic, nothing but it can cause the disease, pneumonia disease to the mice, and it is capsulated. You know that the capsulated forms of bacteria are pathogenic and non-capsulated forms of bacteria are non-pathogenic. Here our S3 strain is a capsulated form so that it can cause the pneumonia disease to the mice, so that it is virulent or pathogenic strain. Here this is the S3 strain. This is the S3 strain and this is the capsule of S3 strains. These capsules are made up of mucus polysaccharide. Because of this mucus polysaccharide, when we grow this S3 strains of Diplococcus on a medium, they will give a smooth and shiny colonies. Smooth and shiny colonies. Here S stands for smooth, and let us move on to R2 strains. Here R stands for rough, means they devoid of a smooth capsule, and these R2 strains are avirulent or non-pathogenic, means they cannot cause the disease, pneumonia disease to the mice, and these are non-capsulated. And when we grow these strains of Diplococcus on the medium, they appear as rough colonies. Let us come to know the way in which Griffith has conducted his experiment. The main aim of a Griffith experiment is to know which strain of Diplococcus pneumonia is pathogenic and which strain of Diplococcus pneumonia is non-pathogenic. Okay, so in his experiment, in the first step, he took live R2 strains. These are the R2 strains. To these R2 strains, he allowed to grow on a medium. On this medium, these R2 strains form rough colonies. To this bacteria, he injected into a live and healthy mice to know whether this R2 strains causes pneumonia disease in mice or not. But in his observation, he found that live mice, the mice is in live condition. This step of his experiment indicates that these R2 strains are non-pathogenic. R2 strains are non-pathogenic or avirulent. In the next step, he took S3 strains, live S3 strains, and allowed to grow on a medium, so that he got here smooth and shiny colonies. Smooth and shiny colonies. To this bacteria, he injected into a mice to know whether these S3 strains of Diplococcus pneumonia causes diseases or not in the mice. But here in his observation, he found that dead mice. The presence of dead mice indicates that these S3 strains are what? Pathogenic or virulent. Pathogenic or virulent. Just this is an observation that S3 strains are pathogenic. Here Griffith wanted to continue his experiment further to confirm whether these S3 strains are truly pathogenic or not. So that he went for the third step. In his third step, he took heat killed S3 strains. Here he took these S3 strains from here and he allowed for heat killing. To this heat killed S3 strains, he injected into live mice, and after that he observed that live mice, the presence of live mice after the injection of this bacteria, heat killed bacteria into it, indicates that these S3 strains are pathogenic. Here these are in live condition, so that they cause a disease and the result is the death of mice. Here these bacteria are heat killed, so that they didn't cause any disease, so that mice is live. And in the next step, fourth step, he wanted to reconfirm that these S3 bacteria, S3 strains of Diplococcus pneumonia are truly pathogenic. This is the observation step, this is the confirmation step, and this is the reconfirmation step. In this final reconfirmation step, he took the mixture of live R2 strains. What he took? He took both live R2 strains and heat killed S3 strains, and he got the mixture of these two and allowed to grow on a medium for a certain period of time. After that, to this bacteria he injected into a healthy mice. After this, Griffith could expect live mice, as R2 strains are naturally non-pathogenic. The S3 strains, though they are pathogenic, but they were heat killed, they are in killed condition. Not only Griffith, anybody also could expect live mice here, but unexpectedly Griffith found that the death of the mice due to cause of pneumonia disease, and moreover he recovered live S3 strains from the medium. Based on this observation, Griffith came to a conclusion that the R2 strains in the fourth step somehow been transformed into live S3 strains by the involvement of heat killed S3 strains. This is the first conclusion that Griffith has got from this fourth step. And the second thing is something that transferred from heat killed S3 to R2 strains, heat killed S3 strains to R2 strains, and so that these R2 strains became live S3 strains. This is the conclusion. S3 strains. Here to this something he called as transforming principle. to this something he called as transforming principle. That transforming principle is said to be as genetic material. Genetic material, as this transforming principle or genetic material is involving in the transfer of a character from heat killed S3 strains to R2 strains. Nothing but a gene responsible for the synthesis of capsule in S3 strain got transferred into R2 strains, so that R2 strains became S3 strains again. Here Griffith was failed to know the biochemical nature of the transforming principle, nothing but whether it is DNA, protein or lipid or a RNA molecule, but he thought that this might be a protein molecule. Thanks for watching this video. Please like, share and subscribe my channel.